Great. Okay. Well, thanks very much for the invitation. I really enjoyed the first two talks of my colleagues. So, so welcome to Miami University of Ohio. It is not Miami, Florida, which I wasn't aware of when I was offered the invitation to come and interview. So I always like to point that out to people. So we're a small university in Ohio. We train a lot of undergrads, but we also have graduate programs. And my research group focuses on polar microorganisms. And I go out to their native environment. I work in a really unique place in Antarctica, which I'll talk mostly about today. But we also bring isolates back to the laboratory. So we kind of have this nice balance between studying microbes out in their native habitat, but also being able to do more focused studies in the laboratory with a number of extremophilic organisms. So the McMurdo Dry Valleys are a unique habitat on the continent of Antarctica. So most people think of Antarctica as a massive ice sheet, but actually there are these special places within the Transantarctic Mountain, which are polar deserts. And I work specifically in the valleys of one of these within the Transantarctic Mountains, and I specifically work on several ice-covered lakes in this region. But I'm also part of a larger group called a Long-Term Ecological Research Site, or LTER. And specifically, this LTER is the McMurdo LTER, and I work with a lot of other amazing scientists. So there's a group of us that are focused on the lakes, but we also collaborate closely with other scientists that study microbes in the soils. We have a stream ecologist, and we have a glaciologist, as well as someone who studies remote sensing. So this project has been funded for over 30 years, and I provided our website at the bottom. We have a great website, which obviously has lots of details about the site, but it also has full public access to 30 years of data. So it's fully available for anyone who, is interested in using our data and designing projects with our long-term data set. So a little bit about how we get to Antarctica. So we fly on commercial airlines to the South Islands of New Zealand. And then we take a military flight from New Zealand to McMurdo Station. McMurdo Station is run by the U.S. Antarctic Program. It's the largest station in Antarctica. And the station itself is basically a spot where my field team prepares. So the station has 1,200 people during the Antarctic summer. A lot of those people are support staff that basically help us get ready to do science in remote places, basically. And this is more specifically how we get to our sites. So our sites are remote, and so we utilize helicopters to fly me and my students out to the dry valleys. And these are some images of us getting ready, flying out to the dry valleys with our gear. And there's a photo in the bottom right of what it's like to hook a helicopter on the lake. So that's standing on top of a lake, and that's me hooking some of our equipment to a helicopter. So lots of fun experiences there beyond science. So we spend a lot of time sort of just getting ready to do science in these extreme places. And here's some images on what it's like to work there. So they are ice covered, but we do have what we call liquid moats. So these are a unique ecosystem that we have liquid water on the perimeter of the lakes. And depending on how warm the summer is, these moats can be quite large and a challenge for us to cross. We typically are working in the middle of the lakes, and we need to drill through 3 to 6 meters of ice with gas-powered drills. So again, lots of other kinds of activities besides science. And then here's a couple of images down on the bottom right of us doing some sampling. So the middle one is some students measuring PAR or light availability through an ice hole. And then the one on the far bottom right is me actually trying to safely sample the moat. So we sometimes we also have experiments studying the unique communities in the moats, and they're kind of hazardous to get to. So this is kind of a way we designed a, peristaltic pump and a long tubing with a weight at the end to sample that ecosystem. So, a few really unique things about the lake. So, they are one of the very last permanently ice covered ecosystems lakes in the world. There are some in the Arctic, but they have experienced a high level of climate change, and now those lakes are ice free in the summertime. So the Dry Valley lakes are very unique from that point of view. They're also very pristine. There's minimal human impact or interactions besides the scientists. They're very extreme conditions, so it's a completely microbe-dominated food web. And the ice cover also has this interesting effect of a highly stratified aquatic environment. And on the right is a little cartoon of just generally what that stratification looks like. So again, there's a permanent ice cap on the lakes. The upper water column where there's light or the photic zone, it's typically highly oligotrophic, so very low nutrients, excessively high oxygen, and then we have a permanent chemocline. And below the chemocline is often anoxic, no little to no light, and some of the lakes are hypersaline. So the organisms there experience multiple stable extreme conditions, which my lab's very interested in. And then last, another fascinating thing for people that are interested in photosynthetic organisms, it's 24-hour daylight in the summer and 4 to 5 months of darkness in the wintertime. So, the life is harsh there, and so life is small. Life is all microscopic. There are a number of interesting different ecosystems in the dry valleys, and my lab specifically focuses on the microbial eukaryote community or the protists. And these are some images that a student of mine captured of the wide morphological diversity of these protists. In our lakes. And so these protists or microbial eukaryotes play really outsized roles in the food web. So as I mentioned, the food web is completely microbe based. There's not even any like aquatic invertebrates in the lakes. And so we've been working on building this microbial based food web and understanding it for a couple of decades now. And this is kind of our current view of the food web, including these arrows are showing interactions between different microbial components of the lakes. And so the green arrows are showing positive interactions. So, for example, we're studying interactions between photosynthetic protists and heterotrophic bacteria. And then the red arrows indicate negative interactions. And so for example, the top predators in our ecosystem are predatory and parasitic protists. And so they predate on heterotrophic bacteria, but there's also some interesting things like algal parasites, like this image of Personia, for example. Another interesting group we have are constitutive mixotrophs. So these are protists that are capable of photosynthesis, but they also have they're capable of predation. So they have a simplistic digestive system, and so they can both, interact positively with bacteria or negatively as predators. And just to kind of spend a couple minutes talking about the other kind of work that I do in my laboratory. So, as I mentioned, we have a number of isolates, and these are two of the isolates that we've studied in my lab. So, one of the things that's really um, a great advantage of these lakes is that, as I said, the lakes are chemically stratified. And so we've isolated organisms from the same lake, like these two that I show here, that are uniquely adapted to the layer of the lake that they were isolated from. And so these are two Chlamydomonas species. So Chlamydomonas is basically a single-celled plant. So it's a very good model for studying processes of photosynthesis and other plant processes. And we have studied this organism, Chlamydomonas priscui, for a couple of decades now, and it's really become a model of cold adaptation for photosynthesis. So it was isolated from the deep photic zone. Obviously, it's low temperature adapted, but it's also adapted to high salinity. So that's a really extreme combined stress for a photosynthetic organism to deal with. And more recently, we isolated a second Chlamydomonas, which we named Chlamydomonas ice MDV. It is from higher in the water column. It doesn't have the same um, high salt tolerance, but it has a unique physiology, including uh, a lot more plasticity in its physiological responses. And so this slide, which I don't have too much time to spend on, but it's basically um, summarizing a culmination of 20 years of work in Chlamydomonas priscui. And it has some really fascinating physiology, and we're still working on this organism. So some of the things that we've discovered in this organism. So on the upper left here, it says no state transitions. So state transitions is a very conserved response across all photosynthetic organisms to rapidly redistribute energy, redistribute light energy. And our organism, C. priscui, also called UW241, totally lacks this common response, probably because it's adapted to these very permanent extreme conditions. On the other hand, it has really um, good tolerance to environmental stress, such as the combined stress of low temperature and high light. And so that's what this figure is showing. So on the left is a typical temperate mesophilic species, Chlorella vulgaris, under, sorry, the label got a little messed up, but under um, low light and high light under low temperature stress. And you can see the organism is very stressed. It loses its chlorophyll. The Antarctic organism doesn't bother it at all, just basically looks the same, keeps growing, can actually grow faster under that stressful condition. And down here is basically some data about the photochemistry or photosynthetic health of the organism. So basically it's showing that it keeps all its chlorophyll, it can grow faster, and this is a measurement called excitation pressure. It's under much lower excitation pressure. And one of the reasons that it's capable of this is that the organism has constitutively low reactive oxygen species. So this is a kind of a relative measure of ROS here, comparing, again, a temperate species, SAG 4972, with our Antarctic organism. And basically, regardless of stress, it has very low levels of ROS. And we've gone on to characterize this, and it has a really constitutively high activity of the ascorbate peroxidase pathway. And then in the last minute, I'll just mention the last finding that we're still characterizing today. And that's these two figures. So this one up here shows novel phosphorylation patterns. So the Antarctic organism has a completely novel example of phosphorylation. So it has underphosphorylation of some proteins, and it has overphosphorylation of novel proteins. And we think that this phosphorylation is related to the organism being able to assemble this membrane supercomplex, specifically photosystem 1, Some of these novel proteins are part of this supercomplex, and the assembly of this supercomplex allows this organism to drive high rates of an alternative electron transport pathway called cyclic electron transport. And this pathway is related to, again, constitutive stress protection and the ability to maintain high rates of ATP synthesis. So we think this is also, all of these constitutive adaptations are really important for this organism to survive high salinity and low temperature. And so to conclude, the McMurdo Dry Valleys are natural laboratories to study questions around microbial ecology. The protists play outsized roles in this ecosystem, and the MDV lakes are genomic reservoirs for novel extremophiles. So thank you very much, and thank you again for the invitation.